Okay, today we're going to try to get through the other basal transcription factors. You remember basal transcription factors are the ones that nucleate at the start of transcription at the core promoter. And we've talked about the first to arrive on the scene, the one that escorts tau binding protein, which is TF2D. We've talked about alternate forms of TF2D, the SAGA and the PCAP, and you understand that you need to know what the core subunits are, the ones that look like, sort of mimic the eukaryotic nucleosomes. That would be 4, 9, 6, 12. So now we're moving in the progression of how these things continue to nucleate, the first being TF2D. What comes next? And we've talked about TF2B, so that's usually the next, although we don't know whether it's really B that comes before A or A before B. In vitro, when you try to reconstitute in vitro transcription, it doesn't seem to matter which you put there. So it's B or A or B, D, B and A. And then the next to follow are as follows. So what we're looking at is the pre-initiation complex fully assembled. And here you see F. F usually is tightly bound to the RNA polymerase itself. So when F gets there, it's pretty much simultaneously, it's simultaneous with the arrival of RNA polymerase, since F is so strongly attached. And B has connections to F, and F, of course, is attached to the polymerase itself. So let's see what the first slide here is. This is the Mr. Potato Head model. And sometimes I put this on exam, and when this is all put together, I'll have an arrow going, what is this yellow thing? Of course, that's the N-terminus of B. This is the C-terminus of TF2B, the 2 cyclin domains, and goes underneath the DNA, and that's the N-terminus. This is kind of a crude representation. This is F in blue. We haven't talked much about F, but it's spread out across the complex. It has conservation with some of the sigma domains 2 and 3. That's what the 2 and 3 designate there. The others, which is where we're going today, would be, okay, F is recruited by B, and then you have E and H. And E and H usually come together. E helps assist in the functions of H. So let's see how this works. Okay, so apparently we're going to take a couple of detours before we get to that story, because there's a very important story about the carboxy terminal domain. It is involved in triggering the transition from initiation to elongation. You know, this is kind of what I like to emphasize with the bacterial RNA polymerase. I would constantly say over and over again, what's the event that triggers the transition from the initiation to elongation? Well, it's the same question, but with Paul II of the eukaryote. And it involves the phosphorylation of the CTD of the large subunit of RNA polymerase. It has to be phosphorylated in order to start a cascade of events that eventually ends up in the RNA polymerase leaving the promoter in elongation. So let's talk about the structure responsible for that, or that participates in that transition, and that's the heptad repeats that form the carboxy terminal domain of the largest subunit of RNA polymerase. 2. And here you see the repeat structure here. It's 7 amino acids, 52 in mammals, about 42 in plants. Some of them are not completely conserved, so it gets to be sort of a judgment call where you say the consensus is broken down and whether it's truly a repeat. Yeast has 26. If you truncate this in yeast, you'll find that it's very dislabeling. These cells eventually don't make it anymore. It must be unphosphorylated or hypophosphorylated in order to join the pre-initiation complex, PIC. So in order to arrive at the promoter, the CTD of the polymerase is under or hypophosphorylated. And then as all the cast gather there and the complete pre-initiation complex is formed, then you get a transition, a phosphorylation event where you begin to phosphorylate as many of these repeats as you can to finally, it loses affinity for the pre-initiation complex and is able to sort of disassociate from that and go into elongation. Mediator complex. We haven't talked about mediating. You may or may not have heard, maybe, I suspect nobody's heard of mediator as a transcription factor in plants or in all eukaryotes. Mediator did not come from archaea. This is a eukaryotic invention. It's a very large complex of proteins, and it's involved in transcription at every Pol II promoter. It's an essential component of the transcription complex. It's called mediator, first discovered in yeast, and now it's confirmed everywhere. And we've cloned some of the subunits in plants and worked with them in my lab also. So the hypophosphorylated CTD will bind to mediator in addition to the other components of the pre-initiation complex. It has several phosphorylation states, the CTD. Starts off with no phosphorylation, and the first thing to happen is there are 2 serines involved that pick these that participate in these phosphorylations. It's serine number 2 and serine 5. You'll need to know specifically, there are only 7 amino acids in the heptad. So position 2 is a serine, position 5 is a serine, and both of those are specifically phosphorylated. The first to be phosphorylated is 5, and then you have an intermediate situation where you have 2 and 5, and then finally end up just with 2. So it's sort of an easy transition to remember, except you have to do it backwards. It starts at 5 and counts down, right? 5 is the first one, then it goes to 2. Okay, there are different kinases involved at these 2 positions. The first is CDK7, and this turns out to be a subunit of the TFIIH basal transcription factor. So this is a role for H, one of its several roles. It provides the kinase which starts the ball rolling in putting a phosphate on serine 5, and it does it on as many of those repeats as it can, but always at position 5. That in turn prepares the way for the binding of other things, which would not bind unless that was pre-phosphorylated. So in the first 2 steps here, the RNA polymerase is still at the promoter, and this phosphorylation of 5, serine 5, is the trigger that allows it to transition into elongation. And then in that transition, there is a second kinase. It has several names, PTEFB. I'm not sure what the P stands for. Polymerase transcription elongation factor B is pretty close. I'm not sure about the P. But PTEFB is a name. It's also called a CDK1. That does position 2. And eventually there's a phosphatase that removes the 5 prime. So you start with the 5 prime, you put on a 2, and then you eventually take off the 5 and leave the 2. Ultimately, you'll end up with removing both of those as it now circles back around to reinitiate. It has to be hypophosphorylated with the phosphates removed from both of those key positions as it rejoins to reinitiate at the promoter. Back to building. So we talked about D and the different forms of D, the PCAP and SAGA. A and B come, and the next thing to arrive is F, and F is stuck on RNA polymerase II. And what brings F there? There's that zinc ribbon at the N terminus of TF2B. Not only does that attach B to the wall of the RNA polymerase itself, but initially it's out there making contact with the small subunit of TF2F, RAP30. So it's involved in recruitment of F, which brings the polymerase, and then once it gets there, it comes off of F, and now this is where it begins to snake down into the interior of RNA polymerase. And then the last two are E and H. And now you know the full cascade of events to build the pre-initiation complex, except for one important member, which is mediator, and we'll talk about that a little bit separately. So it's not an impossible thing to know. D, then A and B, F, which brings polymerase, and followed by E and H. So let's go through some of the players here. This is F. Remember, it has 2 subunits, a big one and a small one. RAP 30 and 74. RAP stands for RNA polymerase associated protein. Actually, 2 of TF2Fs get together to form a tetramer. So you see here, it's as a tetramer when it's riding along with RNA polymerase. And you see down here this crude drawing of TF2B. That zinc ribbon at the N terminus of TBP, initially it's used to make contact with RAP30, involved in the direct recruitment of F and the polymerase itself. So what does F do? Well, it serves as an attachment site for its own recruitment by B. It reduces nonspecific DNA interactions with the polymerase. Initiation and elongation. So we have kind of vague ideas about what it does. You can see there's not a lot of something you sink your teeth in here. It decreases pause and protects elongation complex from arrest. It keeps things moving along. But that's about, we don't know a lot in terms of the details of mechanism with F. Okay, but it does play a role in this phosphorylation cycle that you'll need to know about. You remember we phosphorylate serine 2 of the CTD, and that's done by H, CDK7, and then we phosphorylate position 5, and then we take off the phosphate groups. Well, the phosphatases that remove the phosphates, those phosphatases are recruited by F. So F is responsible for getting rid of the phosphatases. So F is sort of working against some of the early events, right? F is working against the action of H. H is trying to initially put a phosphate on every serine 5, and yet F is there going, how can I recruit a phosphatase to take those things off? So it turns out that when it's all together in the pre-initiation complex, F is prevented from acting. it cannot recruit the phosphatase when it's stuck there in the pre-initiation complex. It's only when it frees itself from the promoter and rides along with the polymerase still that it's now unmasked and able to recruit the phosphatase. So it does not take the phosphates off. It recruits a phosphatase that brings it back down to the hypophosphorylated state. So what is the signal, the button that has to be pushed so that it goes from not able to recruit a phosphatase to now able to recruit a phosphatase? It's the contact with TF2B. When it leaves the pre-initiation complex, it's no longer touching TF2B, and that now unmasks the domain that allows it to recruit the phosphatase. So F is recruiting the phosphatase, preparing it for the next round of initiation. That's all you need to know about F. Does F go back to archaea? My last review of the literature did not indicate that. But, you know, things always change. E goes back, B goes back, TBP goes way back, but F and H, don't think there's evidence that they go back to archaea. Okay, E, this is one that does go back. The large subunit goes all the way back to archaea. So that would be the alpha subunit versus the beta. The 57 kilodalton one was there in the beginning. It normally is tightly associated with H, and they sort of positions H and participates in a lot of the functions of H. So it's shown here with E going first and then H. You can make it happen that way in vitro when you try to reconstitute a pre-initiation complex, but is that the order in vivo? Hard to say. Modulates H, and these are the things that H does that E helps modulate. It's helicase activity, ATPase, and kinase activities. In other words, everything H does, it does it better because E is there making sure it's positioned correctly. Okay, we're at H. Pretty soon we'll be able to go home, right? This is like TF2H. I'm sorry. No, I'm not sorry. Yay, we get to stay out of the sunshine here and look at pretty slides. H, 9 subunits. You definitely will not have to memorize well, you may have to know most of them, but not every one. Really, that'd be just ridiculous, right? Ridiculous. And I hope one of these little spinning signs that says not on exam 4 does not pop down here. That just ruins the whole thing. Okay, here we go. 9 fantastically interesting subunits of TF2H. It's great to be alive today, is it not? Well, look at that. It finally joined the pre-initiation complex, making contact with E. Okay, the first thing it does, promoter melting. We go, why do you even need it? Because TF2B has a B linker domain that's mimicking the way sigma melts the promoter. What do you need this guy melting your promoter? In fact, TF2H, remember, has 2 helicases, one bound to each strand of the bubble. Then you go back and you go, wait a minute, melting's not the same thing as unwinding, is it? TF2B melts, but it can't unwind other than just a melt. It can't translocate that bubble. The helicases extend the melt to form a full transcription bubble. You have 2, a 3' to 5' and a 5' to 3', which is, of course, they will have opposite polarities if one is bound to each strand because the strands have opposite polarity. So promoter melting. This is over and above, actually the melting, it's actually extending the transcription bubble. True helicase activity. Promoter clearance by its kinase activity, and that's the ability of the kinase, that's one of its subunits, to phosphorylate serine 5. And then it'll lose the kinase and its controlling protein, the cyclin. Those will come off, and other subunits dedicated to nucleotide excision repair will join it. So the core of TF2H is simply those two helicases. That always remains. If it's involved playing a role in transcription, it'll have a kinase, and kinases are always controlled by cyclin, so it'll have cyclin H. That's hard to remember. Cyclin H is a part of TF2H, and then CDK7. Now, how do you remember that 7? Because we're going to talk about CDK8 also. I can't remember 8, but I can remember 7. This is the way I do it. H, heaven, 7. Heaven, 7, and that gets the 7 out of the way, and what's its cyclin is back to H again. So there you go. You can't, it looks like maybe medical school is a possibility now that I told you that. Mutations, of course, in these core subunits, the two helicases, and a few more, and it's very hard to predict, but you've seen these children with the small heads and they look like old men, Cochrane's syndrome. They have mutations in their helicases of TF2H, which sort of seems to imply that part of aging is just inability to repair. And I'm sort of looking at me going, I don't know. So then I get back to thinking about the Faustian bargain that multicellular life made. Like if you have a pool of bacteria, they don't really have genetics, but micro people think so. They're dividing, and everyone is being tested. If you're not fit, if your genome has picked up mutations, you don't make it. But the multicellular organism, I got mutations everywhere, and yet I'm still sort of making it. That's one of the trade-offs of being multicellular. You only go through the test where you drive the car around the block, check the lights, honk the horn, turn the radius, the air conditioning on, see whether the top goes back and comes up or not. Only doing embryogenesis. You pass that and you go, I'm done. And then these whole group of cells just go together like a big colony stuck together for, 50, 70, 71, I hope, 3, 4 years. But a lot of them don't deserve to be there because they picked up mutations, but they're a spleen cell. I hate to berate the spleen all the time. It's mainly because I don't understand spleens. And that causes a lot of misunderstanding sometimes. It's just lack of communication. It never talks to me. I don't know whether I really have one or not. I don't know if I've offended or not. Is it me? What is me? What are you? Let's keep going to the next slide here. Semester's rapidly coming to a close. Okay, the core. That's the two helicases. There they are. And the other module that is there, the one that's switched out when it goes from transcription to nucleotide excision repair, that's where the kinases are. In fact, that submodule of H, yeast people are strange, they've given it its own little name. They think that it's TF2K. What do they know, right? We think it's called, I think that's a kindergarten word, I'm not sure, CAC. Really? So you can call it TF2K, or you can embarrass your entire family and call it CAC. That's what I learned in college. That's where the kinase is. CDK activating kinase. And which kinase is it? No, H, heaven, 7. It must be CDK7. Which cyclin controls it? Cyclin H. There's that little CAC module. It's got cyclin H controlling CDK7, and it's got one structural protein to hold it together. And this is called the CAC module, and it turns out this module has a day job. In fact, most of the time, it's not in association with TF2H. It's phosphorylating stuff in the cell cycle. It's controlling the progression of going through G1 and S and G2 and all that. That's its main job. Free, floating, not associated with TF2H. So this seems to be sort of a contrived, cobbled together afterthought of transcription, doesn't it? It's taken things that have roles in other processes and joined them together. Anyway, that's our view now. Aha! It was so simple. I brought up the M word again. Mediator this time. Mediator. It turns out this cyclin H, you remember the cyclin is the regulatory unit of a kinase. Well, there is something that controls the controller. It turns out that mediator, this thing we haven't talked about yet, has a submodule itself that has a kinase. That kinase targets the regulatory subunit of CDK7. So mediator can go in and phosphorylate cyclin H, which now means that it's inactive. That TF2H no longer can phosphorylate serine 5. This is a way mediator can shut down transcription. Just reaches in, phosphorylates the controlling subunit, the cyclin H, so that it cannot direct CDK7 to phosphorylate the CTD. And that little story you'll need to know about how mediator can repress or inhibit transcription by interfering with the activity of H. Okay, so here you see TF2H with its core, the 2 helicases, and this CAC module. And the CAC module is involved in phosphorylating the CTD, but CAC, when it's independent, is involved in regulating cell cycle, apoptosis, those sorts of things. There's a whole nother module, these XP proteins, G and so forth, that come in and take the place of the CAC module when the complex is in a nucleotide excision repair mode. So the core participates in both. It just has 2 groups of submodules that it hangs out with. Okay, so I think this is a multiple choice question on the exam. I'll say, which of the following is not a function of TF2H? There's a whole bunch of functions of H. You have to pick out the one that doesn't fit. So here we go. It phosphorylates the CTD. Okay, we talked about that enough. Together with E, it melts or performs helicase activity at the start of transcription. 3, this is one we haven't talked about. It's required for the formation of the first phosphodiester bond in transcription, the hardest nucleotide. is, well, the hardest two, or the first two. Joining, putting the first one there without a template, without a primer. It has a template, without a primer, and then joining the next one to it. The first phosphodiester linkage is the toughest, and H facilitates that. Okay, so that's not a long list. So let's look at, to me this, because I got this thing I'm running from, it's a diamond ring. To me, TF2H is a diamond ring. Back to these M words. And the two helicases form the two sides of the donut. So this is the core of TF2H. And this little knob here is this dissociable CAC. This is where the kinase is. This is the state of the art until a couple of years ago. And so what I've done is I've taken more recent X-ray crystallography blended with EM topology. It's gotten to the point now, you know, where you can measure with EM, electron microscopy, you can see the outside contours of molecular complexes. You can see the bulges and the valleys, but you can't look inside and see anything. You can't see ribbons or anything. So we have been able to blend that with software, with the X-ray crystal structure of where there should be bulges and dents because of where the helices and beta sheets are. We've combined that to construct very accurate and revealing pictures of how the substructure is reflected in the outside topology of molecules. And so with that, it really has advanced a very detailed understanding of structure, and from structure follows a more rational understanding of mechanism. Well, that's kind of the opposite sign, isn't it? It's not twirling, it says know this. Okay, we're going to get, this is where I'm going to splice in the slides that when I did an update of transcription. So, but we'll get through this story and then I'm going to show you what TF2H really looks like. So now we're just going through some principles of transcription. Multi-step assembly versus holoenzyme hypothesis. We haven't talked about mediator yet, but we will. Just take my word for it. It's a large complex that is going to be sort of wrapped around everything. And we know it's involved in repression because it can modulate the activity of H. But it's also required for activation, participates in activation also. So we'll get to that in a minute. But one of the big revelations was that originally people thought it was just the basal transcription factors, everything but mediator. They didn't know there was a mediator at all. And Kornberg's group in yeast discovered it and gave it a name. And what they found is that most RNA polymerase and TF2B and all these things, well, not TF2B, but E and H, most of the members of the pre-initiation complex were already preformed into one giant megacomplex. And it wasn't recruit TF2D first, and then here comes A, and then B, and then F, and it brings polymerase. Everything was hovering over like a giant mothership looking for a promoter. And that was called the holoenzyme complex. And so there were really two steps. You D, and then everything else came in a great big conglomeration. So the opposing hypothesis, the original hypothesis, was multi-step assembly, where each little basal factor comes in a particular order. And that was put together by many researchers in many labs, lots of graduate student, PhD dissertations, where in vitro, we could recapitulate the forming of the pre-initiation complex by adding very highly purified components one by one so that we could make transcription work specifically. I knew some people who were trying to do this in plants. It never was possible. It burned through several really outstanding careers. The plants have this cell wall that really blocks access to the inner workings, and that just technically just too difficult. Anyway, multi-step assembly. First thing to happen is a transactivator that's bound outside of the core promoter will recruit TF2D, that's TBP plus the TAFs, or one of these alternate complexes. The next thing to happen is either B or A comes, but eventually B and A, they decorate each stirrup. So now you get this big half donut circling the start of transcription. And then once you have the DAB complex there, then you get recruitment of the other basal transcription factors. F is going with polymerase, and E and H will go to polymerase also, but usually after the polymerase has arrived. Multi-step assembly. Once they all get there, all the reactions are driving assembly, but as soon as H gets there, H begins to work against assembly, right? As soon as it phosphorylates serine 5, that's working against assembly. It's making the CDT lose affinity for all the other members it makes contact with in the pre-initiation complex. And so the RNA polymerase with F depart. Transcription cycle, this is what we've talked about. And this is the role of the phosphorylation state of the CTD. Now we're beginning to talk about mediator a little more. I've drawn in now, it seems to be a big pale green blob. We'll learn much more about its structure in a minute. But the mediator itself has affinity for the hypophosphorylated state, and it loses affinity. The tail, the CTD, comes off of mediator and TBP. It loses affinity. This is showing this tail, the CTD, coming off of mediator as it's being phosphorylated. And this is what we've talked about. And this is talking about the role of F. F is prevented from recruiting the phosphatase by its contact with B when it's actually in the pre-initiation complex. And then once it's totally hypophosphorylated at the end of transcription, now it can reassociate with things. It can now reassociate with mediator, and the two together now can ride back to the promoter along with F. What triggers the transition from initiation to elongation? Phosphorylation of RAP74. Nope, sounds like I'm saying stuff, doesn't it? But I'm not. Phosphorylation of the CTD by PAL2 by H. That looks right. Let's look at some more here. H3 by TAF. D. Nope, it's got to be 2. Okay. I think this is the last of the basic concepts before we look at some pretty pictures of structure. So we've gotten through the series of events where we've recruited and nucleated a pre-initiation complex. We've gone through a series of phosphorylations that allow the polymerase to escape. What happens to all that beautiful pre-initiation complex that took so long to assemble? You know, a eukaryote, you mainly have convoluted structure. You've got regions that are condensed and regions that are open. It's a tangled mess. Seems like to me it takes a while. It's difficult to find a promoter and get everybody there at the same time to form the pre-initiation complex. It'd be really crazy once you spend all of that time building the pre-initiation complex to destroy it and have to start all over every time you want to reinitiate. After all, specificity was the advantage of having a complex assembly cascade to make sure that it's this particular promoter under these particular conditions that you want to be active now. But there's not much advantage once you've done that to having to go through that whole assembly process. So it turns out that most of that stuff remains at the promoter to really speed up the reinitiation process. So let's look at that structure. It's called the scaffold. Now, the proteinaceous core of a chromosome is called a scaffold. This is another structure that's called a scaffold. It's all that stuff left behind at the promoter after the RNA polymerase has begun to elongate. And I'm going to ask you what stays behind. So let's see how this works. Here you see mediator, the polymerase, you've got E and H and F, E and H and F. All of this is TF2D with TBP being a prominent subunit. Got A and B, and you've got D being recruited there by a transactivator. What happens, what stays behind? 3 components apparently. Well, F certainly leaves, right? Because it's got this romance there with Paul II. What else? B? Really? B? You're going to go away? To B or not to? So F goes and B goes. I'm afraid to push this. Well, 3 components, I guess, is RNA polymerase is the other one. I said, what's the third one? F and B go. That's why I was going, I wonder what we're going to find next, because I didn't know. There's nothing next. This is it. It's F and B, and then Paul III being the 3rd component. So that's pretty handy, right? Everything now is here. It took so much trouble to assemble it. B goes. You know, B is snaked down into the active site of RNA polymerase, so it may not be too surprising that B might be totally dislodged and upset with the whole thing. His feelings hurt when the polymerase runs off with F again. Well, c'est la vie. That's the way it goes. This is the evidence for the scaffold. So there's nothing new here. This is just what these researchers did was to take yeast nuclear extract, and they have an activator attached to this DNA. This is DNA covalently attached. They labeled it with, what's it called? I can't remember the tag now. It escapes me as part of this. My helicases are failing me. They attached the DNA to the beads, and then they started pouring various extracts over the beads to see what would what would nucleate at the promoter. In other words, they were trying to build on affinity beads a pre-initiation complex, and they got it to work. And so the first thing they put there was the GAL4 activator. They put a GAL4 DNA binding. GAL4 is a yeast transactivator, and they have the binding site in the promoter here for the GAL4 activator, and it has an activation domain, so it now can recruit TF2D. They put that in there first, and then they took a nuclear extract that had everything else and poured it over there, and then with antibodies, they probed the complex that was there. And they did it after they'd added nucleotide triphosphates and ATP and so forth. So they see what it looks like before transcription happens. Then they add nucleotides and see what is left. And from this data, we get the information that there is a scaffold that remains behind. Well, the GAL4 activator remains. This is the pre-initiation complex before transcription starts. Everything is there. Then you add the nucleotides. Well, you get the polymerase goes away, TF2B goes away, F goes away, but everything else stays. Even mediator, which you don't know anything about yet. Okay, well that's a convenient place to splice into the next PowerPoint that you don't have. And this won't be the whole thing, it'll just be a few of the, was that it? That was it, yeah. This is the one that you don't have, even though it can be downloaded now for some of you who have your laptops connected this way. So let's scroll through to where we are. This is was a general review of transcription who'd never heard of it before. And so you see these as your old familiar friends. All my lame jokes are still there. It just gets, it's ridiculous. Show me the camera, okay, damn it. Really, it's so sad. Okay, now we're beginning to get pictures of stuff. You've had these pictures? Finally some H. So, instead of that diamond ring, the donut with the knob on it, this is more true to what it really looks like. Rad3, which is XPD, that's one of the helicases. XPB, that's the other helicase. So the two helicases are here. You know, the donut doesn't really close as we thought it did originally. And then this part would be the kinases. Here you see the cyclin H and CDK7, and then the structural subunit there. So sort of things are the same. This is what it looks like now at the pre-initiation complex. Here you can see TBP in green, so it's sitting on top, and behind the DNA on the other side, which if you're sitting on this, transcription's going that way, you see the two globular domains of B, and then the N-terminus snaking down underneath the DNA into the interior. And this is where you're going to have E sitting, right there. E, of course, and on top in the blue being H. Oh, H is massive. It's wrapped around. Blue is H. Here you see from the other side, if you snuck around behind the screen and looked at it that way, you see that. And this thing that's like a big psychedelic porpoise coming up, some kind of drug-induced state, sweeping up behind, or a whale or something that's swirling around in a purple haze. That's mediator, that mysterious friend. Here you have H again. It's right there. You know, I got that wrong. Got it wrong. That purple haze, that's H. See this right here? That's that. See that right there? That's that. The blue is E. Oh dear. Well, I can take it again this summer. The E is blue. Everybody knows TF2E is blue. What am I talking about? And H is obviously a big purple whale or something. Yeah, now it makes perfect sense. Some nightmarish creature in the back. That's H. Look how big H is. Horror. The horror. This is deeply disturbing, these pictures. Okay. Mediator. The pictures. So I'll blend in some of those pictures to what you already have, so there's not much change. But mediator 2 has, its structure is known in much greater detail. But before I go into mediator, I'm going to give you the old version, and then we'll finish with the pictures of mediator, because it'll make a little more sense that way. Which one here? Mediator. So that's not much of a change. Okay, mediator part 5. Notice it looks like a shrimp. This is RNA polymerase itself, and curled around the outside is this big, beautiful shrimp. It has a head, H, it has a middle, M, and a tail. In the fantasy of the molecular biologists, they figured the tail was for grabbing. This is where the activators, the transactivators, they just like a handle, like you pour yourself a cup of coffee. They make contact with the tail. A lot of them do. It's possible to recruit mediator to a promoter by making contacts in other regions, but many contacts are made through that so-called tail component. Here you see mediator 24 subunits. We cloned a lot of those subunits from plants. We didn't know there was a Swedish group who'd worked in yeast formerly who really knew how to do it, and so we were competing against these, and they totally scooped us and blew us out of the water after we spent 5 years. We never published, and we're not bitter. Okay. So you see that the CTD is here, and we have the story about the mediator binds to the complex through its interactions with the hypophosphorylated CTD of the RNA polymerase. It turns out that subunits involved in that interaction were called SRB genes. That stands for suppressor of RNA polymerase B. Yeast people call Pol II Pol B. Pol 1 is Pol A. So this was first worked out in yeast. That's where they learned about mediator. But what they found was that you could put in counter mutations. You could take mutants of SRB, of these SRB genes, and you could overcome here's the first mutant. You take the tail of CTD, the tail of RNA polymerase, the CTD, and you make it shorter and shorter and shorter, and so yeast is very, very sick. And then you mutate that. So you say, is there any gene that can overcome this really short CTD? Because if there is a mutation that can overcome it, that might be a protein that now can bind the short one even tighter. That's one mechanism. You could overcome a very short CTD if you could just grab hold to it tighter. And so those second mutations suppressed the first mutation, which was trimming down the CTD. They overcame the fact that it has such short tails. So they were called suppressors of RNA polymerase B. And that's what the subunits were initially called of mediator, SRB, this and that, and because of this suppression effect they found. This is some of the early imaging of it. We'll see we've got a lot more information about it now. And so this is one view. This is another view. So if you look at the sort of primitive, the shrimp view, these are the different parts. Let's get more. Integrator of positive and negative signals. It means that it participates in transcriptional activation and in repression. We talked about how it participates in repression. This was a yeast-derived model with two hybrid results where they one by ones tested which subunits interacted with which other subunits. And I had a graduate student that did something similar to that approach, but not with mediator, did it with TF2D. But so you see you have these four regions: the head, the middle, the tail, and this thing up here in red. This is dissociable. This comes on and off. This is where the repression activity resides, because this contains the CDK8 kinase 8, CDK8. This is the one that targets H, the cyclin H. This is the one that reaches in and cuts off transcription. It, in turn, is controlled by a cyclin, and it's cyclin C. And I don't know how to remember that. CDK8 and cyclin C. 8 goes in and phosphorylates cyclin H, which controls CDK7 of H. Anyway, that is the repression engine of mediator. And if you have to know one subunit from each section, the one in bold is the one you should be familiar with. The head domain has SRB4. And the exam question is, which subunit of mediator, when mutated, results in a phenotype that is as severe as deleting 93% of RNA polymerase itself? That's a pretty severe mutation. If you delete the subunits of RNA Pol2, that's a pretty severe. You can get almost that degree of severity by deleting SRB4. It's that critical to transcription. How about the middle? We don't know much about the middle, and I don't think there's a question about it, but these MED10 would be an example of the middle. We know more about it now, but we won't need to know anything specific. The tail, though, we get back into a region that we know a little more about. We have RG1, SYN4, GAL11. If you this is sort of the connector to the rest of it. If you mutate RG1, you end up losing the ability to associate the other tail subunits with mediator. So you create a mediator with no tail. And the result is activated transcription really suffers. It's hard to have inducible transcription because many of the transactivators recruit mediator through interactions with SYN4, MED2, MED3, GAL11. GAL11 is another one that's pretty well characterized as an attachment point for transactivators to recruit mediator. Whoop, it's SYN4, not RG1. Misspoke. Not RG1. If you mutate SYN4, that's called the tailless mutant, not a mutation of RG1. And then this is the CDK8, the subdomain that goes in and out, dissociable, that regulates the activity of H. This is an experiment. How can you purify media? And this is how people actually did it. Well, they knew that it bound to the unphosphorylated CTD of polymerase. So why not just synthetically manufacture CTD? Just get a company to make it for you. Just a repeat, 7 amino acids over and over again, and covalently attach it to some beads and pass a nuclear extract over and see if what you get. One of the things you get is mediator by doing that. Here's the second way. How about take a transactivator, a really strong one, and attach that to beads to make an affinity column, and see everything that gets stuck to VP16. VP16, as you remember, that's viral protein 16. It comes from SV40. It's a very strong mammalian virus. But that VP16 acidic activator here is so strong and so universal that it works like crazy in plants and fungi and everything. But if it really does directly recruit mediator, perhaps you'll have mediator stuck to your affinity column, and it turns out to be true. You can purify mediator by either putting the CTD there on affinity beads or a strong activator like VP16. And so here's the other finding. If you use two different kinds of transactivators, VP16, and this is a second one from yeast, you don't need to remember it, but if it turns out that if you do the EM and get the topology of it, you get two different structures. What? Two different structures? That means structure is controlled by what recruits it. So if one of them pulls on one subunit, the mediator has one configuration. If VP16 pulls on another subunit, you'll have another configuration of mediator. Does the configuration of mediator play a part in how it works? What we think is that mediator controls the amplitude of transcription. Not whether it's going to start or not where it's going to start, but how strong the promoter will be. And so how can it, because it's supposed to integrate both positive and negative signals. Well, a simple analog model is simply conformation. What grabs it? Distorts it in a certain way. So how can you integrate that into a theory of amplitude modulation? Well, a simplistic model is that it's either open or closed. Various degrees of being open or closed. That's the conformation aspect of it. When it's really open, reinitiation can easily occur. But when it's in a more closed configuration, doesn't have to be totally closed, but more closed, then reinitiation takes a little fitting and then it can go. A little fitting and then it can go. That simple model explains a lot of it. And here you see that model here. It was, who's the guy? Danny Reinberg. He's really a moving force behind this idea, Reinberg. Okay, so you see this simple model here of its conformation controlled by the regulator that recruits it. And that in turn determines how open it is, which is configuration. And so here you see kind of a grand unification model of how everything might work. It's not, again, by Danny Reinberg. You see TF2D, Pol2, H with the CAC. You got some mediator going on here. Gal4 is a transactivator that recruits mediator. Here, this transactivator actually recruits one of the head module, the SRB4, VP16 interacting with the tail of mediator, GCN4. You don't need to know any of those specific models. That's just there to take up space in your head. This is another name for the cyclin, the cyclin C and CDK8. Its SRB names are subunits 10 and 11. Anyway, you can see how they control the activity state of the kinase in H. So this is what mediator looks like right here. Is that mediator? I think that's mediator. But you see it kind of globs on the outside of RNA polymerase, but we'll see in a minute that it's making interaction with specific subunits. Here we go, exam question. Here you see part of mediator here that's interacting with RNA polymerase B, protein 4 and 7. These are subunits 4 and 7 of RNA polymerase. They are moved by their contact with mediator, and we think somehow that modulates activity of the RNA polymerase. Mediator contacts subunits 4-7, triggers opening of the clamp, provides better access for DNA to enter the active site of the polymerase. So this is another way, this is a more refined model than simply like that. This is saying what it's really doing is controlling how open the clamp of the polymerase is by moving subunits 4-7, making contact with 4 and 7, and how efficiently it's able to move 4 and 7 determines how efficiently it's able to regulate the clamp of RNA polymerase. So doesn't that ring a little familiar? Remember, sigma affects how open RNA polymerase is in bacteria, whether it can, when it binds, it opens it up so sigma can jump and hop and search for promoters. And here you see another more complicated way to regulate the openness of the polymerase itself, the DNA channel. Recent model. I'm not going to ask you to label this because I could not do it myself. But if you just stare at this a while, you'll begin to see, like, that's the DNA. Where would TBP be? You go in red. You go really. B is green. And you're probably going, I don't believe you, I don't care. And this is, okay, how many more we got here so I can show you some pretty pictures? Okay, so I'm going to show you some structure of H in a minute, and I'm going to put those figures on the exam, and I'm going to tell you what to look for there. It'll be very simple. So we've talked about mediator, except I'm going to show you some pictures here. How much time do we have?
About 40 minutes.
40? Okay, good. So this is the last of the general principles. We've talked about now the assembly of the pre-initiation complex. We've covered all of the basal factors from D and A and B and F and E and H and mediator. Now let's step back a little bit and talk about how all this fits in more generally in regulating transcription. How do you get all this to go? Sort of what are the steps in activating a gene in a eukaryote? Remember that a lot of it is involved with controlling the condensation state of the chromatin. And so, as you might expect, there are a series of remodeling complexes, some of which are in your gene structure bioinformatics project. You had to identify some of these proteins, and they had crazy names, and you figured, why in the world did he put those there? Did anybody have anything called SWI/SNF? The SWI/SNFs are different than a HAT. HAT, histone acetyltransferase activity to open the chromatin. These SWI/SNFs move nucleosomes. They remodel the chromatin architecture. They don't remove histone or nucleosomes. They don't modify the nucleosome tails or the histone code. They just rearrange the furniture. Just move the nucleosomes away from the promoter, essentially, remodeling complexes. And the big characteristic on exam is they require ATP to do that kind of work. They don't work for free. The SWI SNFs are remodeling complexes. They don't have HAT activity. They're not HDAC. It's a remodeling activity. It requires ATP to do that kind of work. Okay, let's look back at some pretty pictures. And I'll show you, I'll just think out loud and decide what I'm going to put on the exam right here. You guys can kind of help me with that. Talking to my imaginary cat. Ready? Okay, now you see here an actual diagram of something that's called mediator, but it doesn't look as much like a shrimp. This is a more true reflection of it. You see the head molecule and middle molecule are kind of stacked up on each other, and then the tail is still sort of accessible. That's okay. This kind of helps you understand what's going on over here. H, let's look where TBP is. It would be right here. It straddles the DNA helix, as you can see. And if you're sitting on that, direction transcription's that way, your left foot's going to be there, so that's B. And you can see part of that green going down inside the RNA polymerase itself. And sort of behind it all is this great looming mediator. Nope. That is H. That H is constantly looming in the background, isn't it? Here's H core. Now you're seeing mediator. Just a little bit of mediator here. Oh, we got color. This is getting a little more easy to understand. You have a tail module. This is doesn't look as neat like a shrimp tail as much more strong bend in DNA direction transcription pointed down. This TBP, TF2B here. Then you see that green being the N-terminus of TBP. You've got all of this structure being mediator. And you can see sort of, if you were riding in the saddle, you'd see looming there on your left and going down, like this is you on a roller coaster, pretty soon you're going to go down. The mediator is going on down where you can't see anymore. The pre-initiation complex, this is B, TBP. That's the DNA on the back side, on the C stirrup. You see the green there for B and how it circles, snakes down into the interior of the enzyme. And then on the back side there, you have the hook, the knob, and the beam of the middle part. The head is light blue behind Paul 2 here. If you rotate this around 90 degrees, you could see a little better. The middle is the dark blue. The head is the lighter blue. And you see the middle is really stacked sort of on top of the head. Here you see some of the middle poking out there, so it's behind the head. And here you see that close contact with the head with the subunits 4 and 7. So this is a big monster glob on the outside of the polymerase that always has to be there. Here you see another picture of it. Here you see the DNA. This is a transactivator, and it's recruited mediator here. This would be the tail module, so you see recruitment occurring through interactions with the tail, and then this is this should be the middle again. What's that? Let's go back. Which coloration? The head has the middle has hook, knob, beam, plank. So the middle is behind here, that dark. This is the head part. And yeah, the head part's making contact with the 4-7. The light blue is the head, the middle is the back, and the tail's way back here. So it doesn't have that nice logical orientation that we thought we had it all figured out. And then this is F and downstream, and B is this mucoid green. Here is another picture of it. Here you can see nucleosomes. This is not quite beads on a string. That looks like it's more highly condensed. This is GCN4, which is a transactivator. Transcriptional activation domain is what TAD stands for. And it is making contact with the tail. The tail has two configurations, up or down. Notice here, that's back to the nucleosome structure. This is, anybody remember what TF2K is? It's another name for CAC, except it's in yeast, right? So this is the domain of H that's going to be phosphorylating the CTD. Notice how that CTD sort of snakes through a hole in K, or is that a groove? It's hard to say from this angle. But K has a very close contact, and so if, when the polymerase left, this had to go through K, then that's how it would be able to get each of those repeats. It'd be, which serine is it putting the phosphosome? 5, right? Serine 5. Okay. Let's see. Presented phosphorylate mediator interacts with the tail in the down position. Okay, so the transactivator, when it's down, it's when it can be recruited, except you notice the yeast people being crazy. Down is up and up is down. That's really how it is. That's not confused. That's just your orientation. Can you identify the channel taken by the CTD? So I'm looking for K. There's K. There's H. There's K in the back. Notice there seems to be a little groove, a trough there. This would be a great place for the CTD, the tail, to have to travel through and get phosphorylated as it translocates through the CAC, or the TF2K. And here you see you've gotten so you can recognize this being the tail when it's up and down positions. So that must be mediator. Let's go here again. Be familiar with the location of the major components. So what would I be asking you? I will give you some arrows that I point to when I redo the slide, when you download it, when I have put these together. I'll put big red arrows on things that I consider important. I won't fill the slide with arrows. There'll be maybe 3 places with an arrow. But K is probably going to be one. Okay, this is a detail of the CTD as it's traveling through that CAC domain. This is H. The yellow is the CTD, and that represents the serine being phosphorylated there. Here you see another view of it here. This channel formed by the kinase, or the TF2K, is where the CTD, unstructured, it lies right there. Also, mediator forms one side of the channel, one of the mediator subunits. Okay, what's the time? We still have some time?
You got 20 minutes.
Okay.
We're out of battery.
We're out of battery?
Yeah.
Okay. Just electrons, we can get some somewhere. University's providing us, okay, good. Okay, so we're really making some headway here. When I reorganize this, I'm going to put arrows and make it clear what you'll need to know. I'll update the key terms. I'll put them there. I'll modify the student learning outcomes. I'll try to make it so it's not just lots and lots of confusing pictures. There's one other concept to really bring you up to speed, and you will know more about transcription than anybody in the micro department and most of the people in biochemistry. And that's this, it seems, it's pretty complicated, but it seems at least logical. But there's something that happens with Paul too, every time it seems like. When you start transcription and the polymerase now has left the promoter and life is good and it's happy and it's beginning, it has the basket with the flower petals, it's going to throw them up and go, I entered elongation finally. It stops. It pauses every time. It has to overcome this pause. This is not abortive cycling. That's already happened and finished. Now it's finally going to step onto the main highway, the yellow brick road, and it's got a pause. This pause was not invented by eukaryotes. It goes all the way back to archaea. Some of the things that make it pause are ancient. It's another way to regulate. One reason you might want to pause would be what? If you think back to pausing for the trip operon with attenuation, you got the hairpin loops, that loop 1 and 2, it's the first pause. You're pausing to allow ribosomes to bind. Would a pause serve a similar purpose with Paul 2? Remember, Paul 2's got this crazy CTD that it drags along behind it. Great long tail. Things have to bind it. That pause allows the first thing to bind. Now, what are you going to need first when you're processing a eukaryotic messenger RNA? You're not going to need to remove introns. You haven't made any yet. You're not going to need to add the poly A or to terminate. You're going to need a cap on that nascent end that comes out. The capping complex is the first thing to bind. That pause might be because of that. So I have one slide, not one slide, of course I have a billion slides, but I've got one more file that I'm going to blend in here to give you the pausing story, and you'll know everything pretty much up to date about transcription in eukaryotes. Have any of you heard of this pausing I'm talking about? You haven't gotten this far in biochemistry yet, have you? About transcription? You have? They don't talk about fun stuff. It's always competing with the person beside you. You're hoping they get the B because you're going for the A, and there are only so many they're going to give. This class, everybody can get an A. I encourage it. Just show me some molecular art. That's half a letter grade right there. Now, would a biochemist ask you to give molecular art for extra credit? Just think about those guys. They would? I need to go over there and visit, walk through the halls again. They did? They already have?
No, she wants to do it.
Sure. That molecular art, art's all in the frame. My impression of transcription. It's dark, I can barely see. There are things moving. Okay, this. The pause. Okay, the pause. You know, cats really like their claws. You take, claws make the cat. They always admiring them. Okay, so enough about cats. The pause. So here you see, let's go through the steps. Basal factors plus RNA polymerase are recruited to the promoter. Yeah, we heard that story. And here are serines 2 and 5 in this heptad that's repeated 52 times in mammals, or 50 some. Next step, CDK7, which is controlled by cyclin H, which is a part of that TFIIH, phosphorylates, here's the H, phosphorylates 5, serine 5. And that allows promoter clearance. It actually leaves the promoter and pauses. The PIC formation, promoter clearance 3. In step 3, you get negative factors and the capping complex join. So the negative factors, we don't know exactly how they make the pause happen. Are they anchored to basal transcription? Are they still anchored to the scaffold? It could be as simple as that. But these two negative factors here, DSIF, and the name is convoluted, just know the initials, but DSIF is ancient, goes all the way back to the beginnings of the precursors to eukaryotes, archaea. And it is binding here to the CTD. But of course, its binding depends on its being phosphorylated at serine 5. So the first thing to happen here gets the ball rolling, and then you bring these 3 proteins together. NELF is a metazoan invention. I don't know whether plants have this or not. They might, I'm not sure. But this is the capping complex. Human capping enzyme, HCE. This is the capping complex. I think this is why it's pausing. You've just made the 3' end. It's not flailing around and flapping in the wind. It's easy to find. The little short, stubby, nascent transcript just emerged, doesn't know it's got to get its shots. It gets capped. Well, that's great. It's got a cap. Now ribosomes are going to be able to recognize it. That's a big deal. You really need to put that cap on there, otherwise you might as well not even make the RNA. But now you've got to get rid of the pause machine here. And the way to do that is a second kinase comes in that's not part of TFIIH, and this is the new component here. This relieves the pause. PTEFB. It has several names, but I think it's called kinase, it's K1 or something, let's see. It's on the next slide. But PTEFB, it's the kinase that relieves the pause. So you see, in a sense, H, it causes transcription to start and you get away from the promoter, but it actually makes it possible for the pause to happen because now it's phosphorylated CTD, which is what you need to do in order to get these 3 proteins to bind. The capping complex and these 2 negative elongation factors, NELF, and this has a name related to an inhibitor that it's sensitive to, DSIF. So how does phosphorylation make things better? Well, PETFB phosphorylates position 2. Remember the slide we said PTEFB 2? But it also phosphorylates these negative factors, and it causes them now to lose affinity and go away, except one. There are only two. It makes negative elongation factor go away, but it causes DSIF to change its conformation, and it continues to stay there to promote elongation. We don't quite understand how, but it stays there. But at least it's not causing a pause to happen anymore. Here's another way to look at it. Promoter opening, a transcription factor, chromatin remodeling, these kinds of things. Pre-initiation complex is formed, and once you get the CTD phosphorylated, now you get these two negative factors, and it did not show the capping enzyme here, which they should have, but they did not. And the HCE, which is human capping enzyme, is there. Then you get PTEF. PTEF phosphorylates these position serine 5, and it also phosphorylates the DSIF and phosphorylates NELF also. So let's see if I said it right. Transactivators recruit, transcription, polymerase quickly bound, the nascent transcript quickly bound by these two negative factors, and they're thought to actually bind to the nascent transcript itself, not to the CTD. So I stand corrected yet again. Being wrong is no big deal for me anymore. I'm always wrong. Negative factors bind that negative transcript. Once established, the enzyme capped second serine 2, TPVF leaves, DSI remains, pauses, release quickly. Reinitiation also pauses but is released quickly due to the presence of PETFB, because PETFB is recruited there, recruited by other transactivators, factor 2. So you're not going to need to know the precise details, so what would you need to know? Let's see what the next slide is. This is another paper describing it. Let's see if we can get a figure that's easy to remember. Paused, and now you have the phosphorylation at serine 5. You have the two negative factors, and then you have the PETFB. Of course, PETFB is in complex with another group of proteins. I'm not going to ask you to know that. Then it is recruited by an activator, PTEEF. It phosphorylates the CTD. It also phosphorylates the DSIF. Actually, it becomes a lot of the control in this slide is showing the many ways that you recruit PEFB. You can recruit it through transactivators, and you can recruit it through things that bind to the histone code. And you can recruit it through interactions with mediator, I think mediator. So there are three ways that you can bring this pause relieving kinase to the promoter. Transactivators can deliver it. The markings on the nucleosome tail. This is a bromo domain. A protein that has a bromo domain would bind to the acetylated lysines on the tails, and that in turn would have affinity for TEFB. And here, mediator through several proteins can form a bridge and also bring PETFB to the promoter. So, what's next? What does that last one say down here? Highly active genes. PAF. Is that polyadenylation factor? Might be. Anyway, that's enough. Ways is brought. Here are 3 ways right here. Promoters by specific activators, by marked histones, which bind BRD4, which is a bromodomain, and delivered by affinity to mediator. This is showing you this ancient one, DSIF, and this is where it binds in the pre-initiation complex. Right here, you can see this is it must be B. That's, yeah, B is here, and this is what it looks like in bacteria, archaea, and eukaryotes. There is something that fulfills that function not only all the way back to archaea, but all the way back to eubacteria. And this is something they'll never tell you in biochemistry. Now you know everything, hopefully just before the head explodes, not after. But that's pretty much, you have a pretty comprehensive understanding of the mechanisms of eukaryotic transcription. There is one more little concept that don't need to cover today, but it involves looping at a smaller scale. And that's this transition going from everything else to being a metazoan. It's your cat made the grade, of course. Being a metazoan, we're all fellow metazoans here. We can talk freely about the other organisms, can't quite do what we can do. It's a looping thing, it's DNA looping. We can loop, lupins. And it's, well, I can't help from telling this story. It's all about the TADs, cohesins, the TADs. This rubber band here is cohesin. Remember where we last talked about cohesin? It was in the synaptonomal complex, and it was right there, the central part was the cohesins. It's also involved in chromosome pairing to recognize the sister chromatin. So it's a very ancient protein, cohesins. But it's used in several ways, and this is a new way that metazoans use it. This is the cohesin, but metazoans invented several proteins that can anchor this rubber band at a particular place in the genome, at a particular address. You can form loops that are reversible and programmed to form by these other proteins which can serve as anchor points. And one of those proteins is CTCF. Is that on there? It's not on here right now. So let's step back at what we know about loops, because then we can't appreciate the cohesin CTCF story. We talked about the formation of the mitotic chromosome. We have loops and then nested loops. The protein there is condensin. Condensin 2 and condensin 1. Condensin 2 forms the great big mega loops, loops of 1 million bases or more. Condensin 1 forms the smaller loops, the loops within loops, to form the mitotic chromosome as it condenses. Condensin. This is cohesin. It turns out that there are looped domains, and these cohesins seem to correspond to insulators. It could be the insulator binding proteins in turn bind proteins which in turn anchor cohesion loops. This defines a domain of like transcriptional expression. These are called topological association domains because they are close together spatially. They are clamped together by rubber bands. These cohesion loops form. So that is one aspect of transcription. That's the way that you're able to take regions that are far away and bring them close together and form a TAD. I don't know whether plants can do that or not. Can form the topological association domains over long distances. The ability to form these loops is a big metazoan invention. This is the protein, the new invention. CTCF is the protein that allows these anchors to form. And here you see a consequence of this, forming these big clusters of CTCF cohesion looping. And this is in the antibody gene shuffling, and processing. It occurs in these big nested loop areas that are formed by CTCF anchoring very specifically where to put these cohesion rubber bands. The binding of CTCF, by the way, is blocked if you methylate a GC doublet. So you can control by the methylation state one way. Here you see bringing distal domains close to the core promoter by specifically anchoring the cohesion rubber band with CTCF. And notice what we have here. A TAF also participates in this formation of these loops. There is no TAF3 in plants. And that was when 10 years ago, when we were looking at them, isolating the TAFs from plants and cloning them. We said, there's no TAF3. This is probably why. The plants can't form these long-ranging loops. They cannot control promoters that are 100,000 bases away. 100 kb loops can still be part of a promoter because of this invention of things like TAF3 and more specifically CTCF. End of story. Kettle drums. It's the end. Transcription, more than you ever signed up for. It's too late for drop ad.